Hello, thank you all for joining today. Welcome to the NOAA Omics seminar series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminars from this series are made available on YouTube and posted up on our Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. And at the end, I will read the questions aloud for the presenter to answer. So with that, our presentation today is titled Rebuilding the Coral Tree of Life to Inform the Sustainability of Coral Habitats by Andrea Quattrini. And she can go ahead and share her screen. Andrea Quattrini is a research zoologist and curator of corals in the Department of Invertebrate Zoology at the Smithsonian National Museum of Natural History. She studied the ecology and evolution of corals and associated fauna and often focuses her questions on those that live in the most poorly studied environment on Earth, the deep sea. Andrea strives to work on projects that directly connect with resource managers to help inform conservation of vulnerable marine ecosystems. Andrea has sailed on numerous research vessels throughout the North Atlantic Ocean using submersibles and ROVs, and in 2020 was a Fulbright Scholar to Columbia. She is also passionate about education, outreach, and mentorship, and strives to make science more equitable. Thank you, Andrea, for presenting today, and I'll now pass it off to you.
Thank you, Nicole. Wonderful introduction and for the opportunity to speak with you all today. I have a bit of a echo. Do you hear that? Oh, that's better. Thank you. All right. Well, as I said, I'm happy to be here today to share with you all work that collaborators across the globe and I are doing to rebuild the coral tree of life to help inform the sustainability of coral habitats. I thought I would first start out today with a little bit about my background and how I started studying corals in the first place. I got a master's degree from the University of North Carolina, Wilmington in marine biology, and during that time, I was primarily focused on fishes, studying larval fishes, in fact, but I had the incredible opportunity in the early to mid 2000s to dive in the Johnson Sea Link submersible. I had the opportunity to participate in a research expedition off the coast of North and South Carolina and had the experience of diving in the Johnson Sea Link submersible to a depth around 700 meters below the surface of the ocean and witnessed a cold water coral or deep water reef comprised of Desmophyllum pertusum as seen here in this video. And this experience changed my life. I decided at that moment in time I was going to work on these ecosystems for as long as I could. So I went back to school for a PhD from Temple University and focused on octocorals in the deep sea. And I quickly learned that we knew very little about corals in the deep sea. But what I've come to realize over the course of my career is that we have a lot to learn about corals still. We still don't have accurate estimates of extant coral diversity, and we don't fully understand their evolutionary history. Yet corals are arguably some of the most important animals in the marine environment and perhaps on the planet by their ability to engineer entire reef-based ecosystems from shallow waters to the deep sea. Corals really provide homes for countless other organisms. Well, in fact, around 25% of ocean biodiversity has been estimated to occur in tropical shallow coral reefs. Corals provide numerous ecosystem services, and they provide sustenance to more than 1 billion people worldwide. Now, I'm sure most of you are familiar with these colonial reef builders as seen here, but that's just one part of coral diversity. There's quite a bit of morphological diversity across what we call what a coral is. It includes solitary cup corals as seen here. Some of these are, the image in the middle is actually of one of the smallest stony corals. It's a turbinolid cup coral, about 1 millimeter in width. These are very diverse, particularly in the deep sea. They can be inconspicuous and often challenging to collect. So in addition to these stony corals, there are proteinaceous black corals, and of course octocorals, or colloquially called soft corals, sea fans, sea pens and sea whips, all of which create habitat in areas throughout the marine environment and provide homes to a lot of other invertebrates. Some of these associations can be facultative to obligate and often mutualistic in many cases. So when I'm considering a coral, or all the anthozoan corals, and there's approximately 5,000 species of anthozoan corals. They occur from the poles to the tropics, from the coasts to the abyss at depths more than 6,000 meters. And I'm sure most of us on this webinar today understand that corals are in danger of extinction, although they are sentinel marine species. They are in danger of extinction, and ocean warming, of course, is threatening corals. We've seen in recent years the frequency of bleaching events increase dramatically, where large portions of reefs, we've seen widespread mortality on large portions of reefs, for example, in the Great Barrier Reef, and in the last year off of Florida and the subtropical Western Atlantic. Now, the corals that live in the deep sea aren't immune to ocean warming and climate change. I think of more imminent concern are activities associated with resource extraction, including fishing. These images are a little bit old, they're a couple decades old, but trawl fisheries still happen, and there's a lot of bycatch that can occur, coral bycatch in these trawl fisheries. The image on the bottom is of a diatrace from the New Zealand museum with just a portion of a bubblegum coral. That coral was likely 1,000 years old or more, and you're just seeing maybe a half of it or a quarter of it in that image. Activities associated with oil and gas have the ability to impact corals even in the deep sea, and the resulting oil spills that ensue. We witnessed from the Deepwater Horizon oil spill in 2010 that coral communities and coral colonies were injured by this oil spill. And of more imminent concern is mining minerals from the deep sea floor, and there's a lot of discussion about this now. In some areas that are being targeted, they likely contain diverse coral communities, but we have yet to characterize those areas. Now, I personally think that to mitigate the impacts of all of this change, this climate change and other anthropogenic stressors, really requires fundamental knowledge, including, how many species there are, where these species are generated, distributed, and connected, because we can't protect what we don't know. And I think we need to better understand how changes in the environment, natural changes, and future projected changes can shape that diversity across multiple scales of space and time, because we really can't save what we don't understand. I would argue at the core of this is fundamental knowledge in its taxonomy and an evolutionary framework known as systematics. So we can better document the extent of coral diversity, not just morphological diversity or taxonomic diversity, but functional diversity and phylogenetic diversity. We need to better understand the historical and contemporary drivers of that diversity so we can inform the likelihood of persistence into the future. And so I think it's important to begin rebuilding the evolutionary framework of corals. And, we're primarily using a genomic approach. I think that genomics, as I'm a museum scientist, and I think genomics is really an important part of our toolkit as museum scientists. We can answer such a wide range of questions using genomic approaches, including population genomics, looking at population level processes, to building evolutionary trees and understanding evolutionary relationships across geological time. And of course, using genomics in a comparative sense where we can compare genomes of different species, for example, two closely related species, and understand at the genomic level what might be driving those differences and how those differences are interacting with the environment. So I'm really harnessing the power of genomics and leveraging museum collections, not just here at the NMNH, but all around the world, to improve the taxonomy and to delimit species within several groups of corals and their relatives, the sea anemones. And today I'm going to share with you one story about how we are using genomics to improve taxonomy and delimit species. We're also using genomics to reconstruct the robust backbone phylogeny of the Anthozoan. I'm going to talk about that today. And finally, I'm going to end today with a study that we recently published in 2024, just last month, I think, how we're using genome skimming to build a reference library for environmental DNA and to also harness thousands of additional loci from that skim data. And you can see here, this is just a tree of the two classes. They are classes now, the hexacorals, which include the stony corals, black corals, and the sea anemones, and the class Octocoralia, which includes the soft corals, sea fans, and the sea pens. So, to build this evolutionary framework, I think it's really important to examine some of the challenges that I believe have plagued coral phylogenetics to date, and then we can hopefully find potential solutions to some of these challenges. I'm just going to talk about two of them today. One is morphological characters. So the morphological characters that have been traditionally used to discern one species from another, or to place certain species into particular higher level groups like families or orders, for example. Some of these morphologies can be convergent, meaning that distantly related species can independently evolve the same trait. And that has led us to group species together in a taxonomic framework that are distantly related. Some morphologies are highly plastic, meaning that they can change due to changing environmental conditions. So this image on the right is a study done by David Paz Garcia, where he followed different coral colonies and monitored the changes in the morphological changes in those colonies. Over just a few months, the morphology changed drastically to the point where it looked like a different species. And this was just due to the strength of storms and waves that had come through the area. And finally, some of the morphological characters that we are using to discern species from one another, for example, have really subtle changes. Yet, in many cases, species were described just based on one or two individuals. So we really don't know the full extent of intraspecific variation of some of these morphologies. A second long-standing challenge has been the use of these traditional or conventional DNA barcodes, like CO1, for example. In Anthozoans, there are very slow rates of mitochondrial evolution. This means that there's little to no intraspecific variation. We published a study in 2023 that showed there's strong purifying selection across the mitochondrial genome. And this has led us, in many cases, where we see the same exact DNA barcode in multiple species, and some species that have multiple barcodes. And so really wanted to get at this question looking at these longstanding challenges, I'm going to share with you this story on Sinularia or Sclerophytum and show you how we're using genomics to delimit species within this genus. This genus is highly speciose. There's 190 currently valid species. Many of them occur sympatrically, so they occur in the same reef, the same location, and they're found throughout the Indo-Pacific. Now, species of Sclerophytum are defined by subtle variations in sclerite morphology. Sclerites are microscopic calcitic elements that are embedded into the tissue of octocorals. This is an octocoral here, this Octocoralia, soft coral. And we use these sclerites to discern one species from another. My prior postdoc advisor and close collaborator, Kathy McFadden at Harvey Mudd College, had started barcoding multiple species in this genus maybe 2 decades ago. And this is the tree that you're seeing here this phylogeny is the result of some of her barcoding efforts. This is based on a barcode known as mitochondrial mu-S. Mu-S is a mismatch repair gene that's found in the mitochondrial genomes of octocorals. No other metazoan or animal has this barcoding gene. It happens to be one of the most informative genes along the mitochondrial barcode for discerning species. But as you can see, there is really no, I guess, strongly supported clades across this tree. So all the different colors denote different morphospecies, so species that were characterized or identified by their morphology. So that's in the different colors across the tips of the tree. And all of the different numbers designate the different molecular operational taxonomic units that were defined by some analyses. And what we see is the strong incongruence between morphospecies and these DNA barcodes. For example, if you look at the blue and the purple, they're kind of all strung throughout the tree. Black, S. abrupta, that's seen in the yellow clade and in the red clade. And this kind of result that we see, this is just one example, we see this often in many different groups, not just within the octocorals, but within stony corals, black corals, and sea anemones. And it really led us to the question of whether the observed morphological differences that we see, do they really reflect boundaries between species whose barcoding genes are not informative? Or do these differences reflect intraspecific variation in morphology? And perhaps this is all just one species. I mean, many of them do occur sympatrically and they occur within a big region. So is it just one species? And we sought to answer this question by using a new approach. We also have dabbled in RAD sequencing, which I'm sure many of you know, but we have really been using target capture enrichment of ultra-conserved elements and exons for a variety of reasons. Just to kind of walk you through what this is, very briefly, we designed these baits or probes, which are kind of denoted by these blue bars here. And these baits are designed to hook on to very conserved regions of genomes. But simultaneously, you're able to sequence through more highly variable flanking regions that flank either side of these conserved regions. So we designed these baits and published this paper in 2018. Now, what does this look like in the lab? You basically start with a library preparation, you shear DNA, you amplify it, and then you hybridize these baits to all of the sheared DNA fragments that have been PCR amplified. After hybridization, you amplify them again and you get rid of all of the off-target sequences or sequences that you're not interested in. These are sequenced on a high throughput sequencer. We're aiming for around 2 to 3 million paired end reads per sample. You get the data back, conduct bioinformatics, build phylogenies. You can also call SNPs and use SNP-based approaches, for example, for species discovery and validation approaches. So this is the basic idea of target capture enrichment. As I mentioned, we published this bait set in 2018. This original bait set targeted 1,791 loci across all of Anthozoa. So again, across stony corals, octocorals, sea anemones, black corals, tube anemones, etc. But then we subsequently redesigned these bait sets and published in 2 different papers, a bait set that targets octocorals, so around 3,000 loci, and a bait set that targets hexacorals, and that's around 2,400 loci. And since the original paper in 2018, I think we are really making considerable strides to this idea of rebuilding the coral tree of life. And so far, from what I've counted, there's at least 25 papers that myself or my collaborators across the world have published using this target capture approach to help rebuild the coral tree of life. So to get back at this question of whether these observed morphological differences that we see, whether they reflect boundaries between species or whether these differences reflect this interspecific variation in morphology, we use both this RAD sequencing approach, but also this target capture approach. And here's the results from this target capture approach. All of the different, so on the left is the phylogeny. It's highly supported. At the nodes where you see some bootstrap support values are those that were not 100. So the nodes that don't have any bootstrap support values is supported 100% of the time. All the different colors are the different clades that we see in the phylogeny on the left, and those colors are also seen in the graphs on the right. So the top graph is a discriminant analysis of principal components. Every single circle is a different individual, and again, they're color coded to match the phylogeny. On the bottom is a structure analysis, and all the different colors match both plots. And what we see here is really strong congruence between the genomic data based on these target capture data of ultra-conserved elements and exons and morphologically defined species. There are some cases where we're still not clear what's going on. So in this case, we have a clade, Sinularia penguensis, and a clade that contains individuals of both Slinger's eye and penguensis. We also have a clade of Acuta and abrupta that are kind of intermixed. So to answer this question, the observed morphological differences reflect boundaries between species whose barcoding genes are not informative, and that is a yes. But there are times where there's intraspecific variation in morphology, and we're seeing that in both of these clades of this penguensis and Slinger's eye and the Acuta and abrupta clade. Using these genomic approaches also really enables us to answer questions beyond just systematics and taxonomy, but to answering questions about the processes that are really driving speciation. And what we see here, and just one example, is hybridization that is occurring between this clade of Acuta and abrupta and Sinularia sclerophytum densa. And so we can use these data in myriad ways to answer cool questions about the evolutionary history of corals. Okay, so now I'm going to share with you some, in the next couple slides, the backbone phylogeny of the Anthozoa. So we've established that ultra-conserved elements and exons can be used to delimit species, showing you the case of Sclerophytum sinularia, which is an octocoral. And so now I'm going to share with you results from several papers that we've published based on that original Anthozoan bait set of around 1,700 loci. So here is the phylogeny that we built, again, using UCEs and exons, and it is highly supported. There's lots of different stories to tell, and I'm just going to focus on a few of those today. This tree includes 234 Anthozoans from both major groups, the hexacorals and the octocorals. As I mentioned, this work that we've done has allowed us to revise some taxonomy, and both hexacorals and octocorals are now considered 2 classes within the subphylum Anthozoa. This includes the stony corals or the scleractinians, black corals, sea anemones, both the actiniarians and the zoantharians and the ceriantharians, and it includes a clade of sea fans and soft corals. We were able to revise the taxonomy of octocorals. If any of you are interested in octocoral taxonomy, you can find our paper in the bulletin of the Society of Systematic Biologists that came out in 2022, where we declared two new orders, the Malacalcianacea and the Scleralcianacea. I'm not going to really talk about all the taxonomic changes, but for those of you that are interested, please have a look at that 2022 paper by Kathy McFadden and others. Okay, so we calibrated this phylogeny using 7 fossils, mostly in stony corals. We were able to get a few from the octocorals and also a fossil, one of the earliest confirmed fossils from the Cambrian. And what we found was that the Anthozoa has Precambrian origins. It's been around for a very long time. So estimated mean origin of around 770 million years ago. So Anthozoa has been around for quite a long time. We then used ancestral state reconstruction or stochastic character mapping to map a few of the traits that are really the hallmarks of reef building corals. To determine when they arose in the past and look at patterns of loss and gain within these different traits. So we mapped the presence or absence of the photosymbionts, whether a particular species was colonial or solitary, and then we looked at different skeletal types. And today I'm really just focusing on calcium carbonate skeletons. There are 2 different polymorphs of calcium carbonate that are found across the Anthozoa, aragonite and calcite, and I'll talk about those a little bit in a couple slides. Okay, so I'm just gonna, again, highlight some of the major findings that we found and just share with you, when we see the first gains of some of these hallmark traits across this phylogeny. First, the most recent common ancestor of all Anthozoa was likely solitary. It did not have a skeleton, and it did not have any of the photosymbionts. We see the first appearance of coloniality around 570 million years ago in the octocorals. But again, we see multiple gains of coloniality across the phylogeny. And then closer to the tips, we see losses of coloniality. But this tree, we've expanded on it. We have a scleractinian phylogeny with many more scleractinians in the tree. And we can really see that although there's multiple losses of coloniality within stony corals near the tips, we do see solitary forms giving rise to colonial forms throughout the scleractinia, and it really highlights that coloniality is a highly labeled trait within the scleractinia. We also see the appearance of photosymbionts in the Devonian, and this places this photosymbiosis much older than what we previously knew. However, there is some indirect morphological and isotopic evidence that photosymbioses did occur in Silurian and Devonian tabulate and rugosing corals. Now, for these photosymbionts, I'm just going to focus again on the scleractinians. We do see losses of photosymbionts closer to the tips of the tree, but in contrast to the coloniality is a highly labeled trait. Photosymbiosis, this trait is rarely gained back in lineages after the loss. So it really indicates the potential loss of this function upon extinction of certain lineages. Okay, and now the calcium carbonate skeletons, we see the appearance of calcite within the octocorals, both in 2 different forms, axes. So there are some octocorals where their axes are comprised of calcite, and other octocorals, they're soft corals. They don't have central axes, but they might have these sclerites that I showed you, these microscopic elements that are made of calcite in the tissues. All octocorals do have sclerites, but there are some octocorals that have no central axes, the true soft corals. And then aragonite appears in both complex and robust stony coral groups around 330 to 357 million years ago. Now, throughout geological time, the oceans fluctuated between calcite and aragonite sea states. So we asked whether these past ocean chemistry conditions were coupled with the evolution of those distinct skeletal types. In other words, does the origin of calcite or aragonite, these different calcium carbonate polymorphs, occur when seas favored those conditions? And I also want to note I overlaid two curves, the temperature and carbon dioxide anomalies onto this graph, and those are in red and blue at the top, just so you can see how temperature and carbon dioxide, of course, has changed throughout time as well. So overlaying those intervals of aragonite and calcite sea states, you can see that there is a pretty good correlation between when we see the origins of those different calcium carbonate polymorphs really do match those sea states pretty well. I also want to mention that, 770 million years ago, the most recent common ancestor of all Anthozoa likely didn't have a skeleton. And this was really a high temperature, high CO2 world before the snowball Earth happened. And again, a time that is not conducive to forming calcium carbonate skeletons. So what are we doing now is we're really working towards integrating data from museum specimens, including types, to help resolve taxonomy and rebuild the coral tree of life. We're really unlocking collections through not only target capture, but genome skimming, which we'll talk about in a little bit, and through new imaging approaches, including 3D photogrammetry, scanning electron microscopy, and micro CT. And I'm really working with my colleagues in Australia through Project Phoenix, Kathy McFadden at Harvard Medical College, Santiago Herrera's group at Lehigh Oceans, among others, to again really unlock our collections and help build this coral tree of life. I think that it's really important to resolve what is what along the coral tree of life. I think it's important for informing those making the decisions on understanding what is what and what their distributions are to help protect these vulnerable marine ecosystems from global ocean change. So that's currently what we're working on, and I'm really thankful that museums all across the world are giving us access to their precious type material to incorporate into our results. Which brings me to this slide where I can demonstrate how we are really unlocking our collections in a lot of different ways. This phylogeny that you see here on the left is of some specimens of Passalopora, a stony coral that builds reefs. This work was part of biodiversity genomics postdoc Mike Connolly, and we attempted to get DNA out of our dry coral collection. We're having a lot of success across the dry collection. Altogether at the NMNH, we have around 70,000 lots of corals, and around 30,000 of these are dried. Now, oftentimes dried skeletal vouchers are bleached, but in our collection, we actually have quite a few corals that still have remaining tissue, and you can see that here in all these different images of the Passalopora. Has a lot of tissue on them still. And so we were able to get DNA, viable DNA for genomic purposes for specimens that were collected up to 90 years ago. We were able to anchor these data. We pulled out ultra-conserved elements and exons from these these samples and anchor the data into a phylogeny with contemporary samples. So it really helps us, it's enabling us to make comparisons of of past coral genetic diversity as well. So I think this is just an example that it can be done. We can get DNA out of these samples. We can get enough DNA to put into a phylogeny, and it really is going to open up our collections for lots of different questions that I'm sure I haven't even thought of yet. Okay, so the third story I want to share today is really focused on generating a reference library to support environmental DNA or eDNA analyses. I'm going to talk mostly about corals, but give you an update on other benthic marine invertebrates as well. But when I started here in 2019, I was quickly introduced to the Ocean DNA Initiative at the NMNH. And the Ocean DNA Initiative is a museum-wide effort to leverage our collections to support the use of DNA sequencing to survey marine life and assess ocean health, with a special emphasis on undiscovered dark taxa that may play a critical role in ecosystems. We're developing trusted genetic reference libraries to serve emerging sequence-based biodiversity technologies, helping to unlock the full potential of environmental DNA. But the potential of environmental DNA analysis really effectively lies on the accuracy and completeness of the reference database to which you compare your eDNA sequences to. I know a lot of you on the call here have probably been talking a lot about reference libraries and how important they are. So you can imagine that you have a genetic sequence from an environmental DNA sample. If it's matched to a sequence in your database, you can confidently identify that species as being present in your environmental DNA sample. But if that sequence is not yet available, won't tell you anything. You'll have no idea that that species might be present in your sample. My colleague at the USGS, Alexis Menning, I think she really said it best. She said it's like Googling for something in Google that's not there yet. And that's what this environmental DNA analysis is. It really is dependent upon that comprehensive reference database, the full potential, I should say, of environmental DNA. So we have turned to trying to increase this reference database through genome skimming efforts. And briefly, you share and tag your DNA, you amplify it, sequence it on a high throughput sequencer. I'm really targeting for around 20 million paired end reads per sample in corals, and I'll talk about that more in a second. We get the data back, we conduct bioinformatics. Now this general approach, this low coverage whole genome sequencing or genome skimming, as people have been doing this for quite some time to get whole mitochondrial genomes out of these data, assemble these whole mitogenomes. Again, many of the barcodes that are used for environmental DNA are found on the mitochondrial genome. But what I'm also interested in is bioinformatically pulling out these ultra-conserved elements, RNA operons and other genes of interest from this low coverage whole genome sequencing data or this genome skimming data. So we published a paper in 2024, just a month ago, in Ecology and Evolution, focusing first on corals. Again, we work on corals mostly. So really seeing if this genome skimming approach to assemble whole mitogenomes and to pull out ultra-conserved elements and exons was doable with genome skimming. And so we skimmed samples across a wide range of collection ages, from contemporary specimens preserved specifically for genetics and from museum collections that were either dried or in ethanol. We consider the historical collections here. They were collected anywhere from 1886 to 2022. And here's a picture of the specimen from 1886 that we tried. And here are some of the results. On the left is the number of ultra-conserved elements on the Y that were recovered versus the number of paired end reads on the X. The blue is members of the Octocoralia, and the red members of the Hexacoralia. Every dot, of course, is a different individual. And you can see here around 15 million to 20 million paired end reads. We're able to capture around 2,000 ultra-conserved element loci out of the targeted 3,000 loci of octocorals. And then for the hexacorals, right around 20 million paired end reads is again a pretty sweet spot to get at least 1,500 UCE loci out of the approximate 2,400 loci that we targeted. So skimming works fairly well for again, bioinformatically pulling out these ultra-conserved elements and skipping an entire step in the lab. The actual hybridization step in the lab can be skipped by using this approach. On the right is the mitogenomes that we recovered via just one program. We used MitoFinder, just kind of a quick and dirty assessment of whether we can pull out mitogenomes and whether or not they can be completed or circularized. So on the Y is the number of paired end reads, and on the X are the two different classes, the hexacorals and the octocorals. Whether the mitogenomes were circularized or not are in red and blue. And again, and I think I mentioned that 20 million paired end reads seems to be the sweet spot where we're able to get most of the whole mitogenomes circularized and complete. These two graphs incorporate all of the data, not just the contemporary, but also the historical. And now I'm just going to kind of pull out the historical DNA. On the left is the number of UCEs recovered in that historical DNA across the year. So on the Y is the number of loci, and the X is that collection date. Although in general, there's an increase, right? With the more recently collected specimens, we'll tend to get more loci, but that's not always the case. For example, this specimen from 1886, we were able to get around 1,400 loci, which is pretty incredible. On the right is the number of mitochondrial protein coding genes that were recovered and the ribosomal units on the mitochondrial gene. On the Y and on the X is the collection age again. And it really, age did not matter here. I mean, in most cases, for most of the historical samples, we were able to get maybe not completely circularized mitochondrial genomes, but most of the genes of interest. So this approach is really allowing us to rapidly include taxa to the coral tree of life. This is the phylogeny published in that paper. All of the purple tips are the ones that we skimmed with, we conducted genome skimming on, and we bioinformatically pulled out those UCEs. We combined them with target capture data, and it's really helping us to kind of rebuild this tree at a much faster rate. We're also rapidly increasing DNA barcode data of marine invertebrates in general through this genome skimming approach. To date, just for my lab group, we've genome skimmed 295 species, but we currently have 600 additional species that are being sequenced right now. We kind of did a back of the envelope calculation and found that there are no barcodes for approximately 60% of these species. So we're really bringing these dark taxa to light through these rapid genome skimming efforts. And I want to emphasize that the data are going to be grounded in museum collection data. So the data, when we put it up on GenBank, it'll have a voucher, a USNM voucher number associated with it. And this is really critical to have that voucher-based reference library. So if any taxonomic changes occur, if people come to the collection and want to look at those specimens, that genetic data will be linked to something that is publicly available and searchable, which I think is pretty important. In addition to, bringing dark taxa to light through skimming efforts, we're also bringing dark taxa to light through environmental DNA analysis. My colleague Santiago Herrera at Lehigh University is leading environmental DNA analyses, and I'm really trying to increase the reference database here, but this is a fun story. I just wanted to share it with you. We found a sequence that matched a genus Parasferosclera, which is fun to say, in an environmental DNA sample from the Gulf of Mexico. But this genus was known only previously from South Africa and throughout the Indo-Pacific. So we thought, no way, there's got to be something wrong. Maybe it was misassigned in the environmental DNA analysis, or perhaps just misidentified in GenBank. But then a couple years later, we actually collected this species from the Gulf of Mexico. So, and it represents a species that is new to the Gulf of Mexico, but it is in this genus Parasferosclera. So this really highlights how environmental DNA can be used to find these rare taxa, but how important it is to have a really good comprehensive database. So how is genomics helping us to inform sustainability of coral habitats? Throughout my career, I really think genomics is an increasingly necessary tool for restoration and conservation of corals. It can be used for multiple projects and questions. But what we're using it for is really for that fundamental biodiversity science. It's helping us to delimit species. It's helping us to improve estimates of coral diversity so we can really determine what occurs where and where should conservation and restoration priorities be given. It's helping us to determine what lineages and key innovations are prone to extinction, and also what might diversify under different environmental conditions, and giving us the ability to ask questions about the processes generating the diversity. I will say that Anthozoans have been around for a really long time. They will likely persist into the future, but what that looks like is totally unknown. What we do know from the past is that high temperatures and high CO2 were not conducive to aragonodic reef formation, and they probably won't be in the future either. We're also using genomics to, build that database necessary for environmental DNA, which can help us to monitor ocean health, can help us to monitor marine protected areas, to determine species presence, absence, and hopefully changes in abundance over time. And it can help us to really monitor the effects of restoration recovery, which I think is becoming really more important across the world. There's a lot of efforts, I'm sure many of you know, to restore corals. We have to find out what species have been injured, where do they occur, to what degree are their populations connected, where do we conserve habitat, where do we restore habitat, what are the effective measures of restoration recovery. I think all of these questions really, I think at the core of these answers is accurate knowledge of species identities, their distributions, and their evolutionary history. And it's really a critical time to continue to rebuild the coral tree of life, not only with samples housed in our museum collections, but also with new discoveries. Characterization, basic biodiversity science, continued exploration and characterization of these coral habitats is essential. We need to improve documentation of species diversity and distribution throughout our world ocean. Every time we go out to sea, we find new species, new records, even in what we think are relatively well-explored areas of the US EEZ. And I'll end my talk today with, in addition to this fundamental biodiversity science, it's really critical that we train the next generation of scientists in applied taxonomy and genomic approaches. And we need to continue to collaborate with people across international boundaries so that we can improve our knowledge of coral diversity and help better inform conservation and restoration in a changing ocean. And so I just want to thank all the members of the NMNH Coral Lab and the many fellows, associates, and interns over the years. A lot of people to acknowledge, and my funding sources, including a lot of funding from NOAA OE and the NOAA MDBC program and the Bureau of Ocean Energy Management and NSF. And I'm happy to take any questions. Thanks so much for listening.
Andrea, fantastic presentation. I hope everybody else had some time to develop some questions and also challenge their taxonomic vocabulary today. Thank you so much, Andrea. Great presentation, especially on the precipice of the release of the National eDNA Strategy, which occurred last week by the White House Office of Science and Technology Policy. I encourage everybody online to go check it out. With that, I have some questions to start us off, and then we'll take some questions from the attendees today. So there was a real focus about mid-presentation on integrating new data types to increase genetic resolution, but then also resolution for analyzing, right, these different data types, particularly in regards to genetics. And I was wondering if you could go back and kind of, if you could look, well, not go back, but if you could look into the future and how we integrate these new data types, and how would that impact the genetic resolution that we have of these corals?
Thanks, it's a great question. I think what we're focusing here is integrating actually some old data of these museum specimens. When folks describe a specimen for the first time, or species, excuse me, for the first time, they designate a holotype or a paratype. And so we're really trying to get those sample species into our tree so we know what's been described already, we know what exists and what has left to be described, because there's a lot of confusion in the coral taxonomy as to what is what. Even in like very, even in shallow water systems like the Cropper species complex throughout the Great Barrier Reef and the Indo-Pacific, there's a lot of undescribed diversity there. And so we really need to, again, put these museum specimens into the tree to see what has already been described and what is left. In terms of other data types, these 3D models that we're creating, we haven't integrated, those kinds of data yet into our tree, but we're making those data accessible to people across the world, which is really important for some folks that aren't able to visit our collection here at the museum. We can make that data available, and they can have a 3D model that they can see from their home instead of needing to come to the museum. There's a lot left that we need to do to integrate other types of morphological data into the phylogeny to determine. I showed some traits that we examined, but there's a lot of other traits of corals that we might be interested in, or their habitats or geography where they live, that we can use phylogenetics to try to understand the diversification of those traits or of those species into different regions.
Thank you, awesome. We have a question from Pedro who asks, hi Andrea, great presentation. Do you think new sequencing methods can improve the variability of population genetic studies for deep sea corals?
Absolutely. Yeah, I think we need to continue to explore the deep sea, right? There's entire regions that we haven't yet to sample. And so there's a lot of unknown diversity out there. But I think we also have the ability, I showed you one example of getting DNA out of these dried coral specimens that are housed in our museums, but maybe we can ask questions at the population level through time and how population changes have occurred throughout the past 100 or 200 years or so.
Awesome, thank you. Another question from me. So you had spoke of eDNA, right? And how these reference libraries that are being developed with you and your teams can aid in identifying dark taxa collected from eDNA sampling. Right. But I'm wondering if there is a push and pull or a vice versa benefit of eDNA and its ability, this new sampling tool and its ability to impact these reference libraries. We know that the reference libraries are going to be beneficial to projects utilizing eDNA moving forward. But if we look at it from the reverse lens, how do you think eDNA might be able to impact these libraries? I hope that makes sense.
Yeah, it does. No, I think it's kind of interesting to think about, because I always come from the reference library side of things, but yeah, I think in a lot of cases where we have all of these unknown sequences in our eDNA data, being able to pinpoint what those might be, it may help us guide like what we need to collect more of, right? Like there are a lot of unknown, for example, Medusozoans or jellyfish and other gelatinous zooplankton. They're very difficult to collect and preserve. Maybe we need to focus some of our efforts on those collections. So we find that data in our eDNA, we don't have any reference database, so we know maybe it's a TINA4, for example, but maybe that'll help guide where targeted collections should be, or what groups do we need to better sample. I think that's a great idea, actually.
Thanks for that. Well, with that, we are right at time. Andrea, thank you so much for presenting this wonderful seminar today. Our next one will be July 17th by Catherine Silliman on the NOAA Omics Data Management Guide. So make sure to check that out if you haven't already. Ideally, we'll get this video up on YouTube, or this presentation up on YouTube over the next week. And be in touch with future seminars. Andrea, thank you so much for your time today.
Thanks for having me.
Bye, everyone. We'll see you next time.